Okay, let's dive into a story that starts somewhere you might not expect, a shrimp hatchery, but ends up in a place that could completely reshape the future of medicine. It's a story about a hidden crisis in our food supply and a hero that is, well, it's a virus. So think about this for a second. What if the answer to a massive global food problem wasn't some new chemical or a fancy piece of tech, but something we usually run from? A virus. I know, it sounds totally backwards, but stick with me, because that's exactly what we're going to unpack here. You see, right now, there's an invisible war going on in fish farms all over the planet. It's a microscopic battle against a bacterial plague that's just wiping out one of our favorite kinds of seafood. The stakes here are incredibly high. To really get the solution, you first have to understand the problem. So let's start with section 1, a crisis in the hatchery. Okay, meet the villain, Vibrio harveyi. This is a really nasty little bacterium that causes a disease called vibriosis. And for shrimp larvae, it's an absolute death sentence. In a hatchery, where everything is close together, an outbreak of this stuff can wipe out the entire population, practically overnight. We're talking huge financial losses and a real threat to a major part of our global food chain. So we have our villain, but every good story needs a hero, right? And in this biological showdown, the hero is the villain's oldest, deadliest enemy. So let's get into section 2, the virus versus the bacteria. And here it is. Meet the bacteriophage, or you can just call them phages for short. These are the good guys in the virus world. I mean, they are hyper-specialized little predators that have evolved to do one thing and one thing only: hunt and destroy bacteria. And the best part? They are totally harmless to anything that isn't their specific target. That includes us, and it includes the shrimp. And their attack method is like something out of a sci-fi movie. It's a perfect little surgical strike. First, the phage latches on. Then it injects its own genetic code, basically hijacking the bacterium and turning it into a zombie factory that does nothing but build new phages. And then the final step, the cell just bursts open, releasing hundreds of new little viral soldiers to go hunt down more bacteria. It's just brutally efficient. This is the concept you really got to grab onto. Phages are nature's own precision-guided missiles. Unlike an antibiotic that carpet bombs everything and causes all kinds of collateral damage, a phage is engineered to take out one and only one specific target. All the good, beneficial bacteria around it, totally unharmed. Okay, so we've got a villain, and we've got this amazing potential hero. But who put the two together? Who made that connection? Well, that brings us to section 3, where we meet the expert on the case. Just read the title of Dr. Surekhamol's PhD thesis. I mean, it says it all, right? Her entire doctoral research was laser-focused on this one mission: finding the right viruses, the right phages, to fight this specific bacteria in shrimp hatcheries. This wasn't just some side project for her. This was everything for years. And this just shows you the incredible depths of her work. To solve this puzzle, she couldn't just be an expert in one thing. She had to master phage therapy, understand the deep genetics of both the virus and the bacteria, know all about the biology of the shrimp themselves, and be an expert on how pathogens move through water. She had to understand every single piece of this. So with all that expertise, with all that focus, there's really only one question that matters. Did it work? Let's find out. Section 4, the research in action. This timeline just blows me away. It shows such incredible dedication. We're talking over a decade at one single institution, the National Center for Aquatic Animal Health, just systematically chipping away at this problem, moving up the ranks from a junior researcher all the way to a postdoctoral fellow. This wasn't some flash in the pan. This was years and years of hard, focused work. And then, here it is, the payoff, the scientific proof. In 2013, she publishes this paper in a major international journal, and the conclusion was just crystal clear. She had done it. She had successfully found phages right there on the shrimp farms and proved in the lab that they were incredibly effective at killing Vibrio harveyi. But hold on, it gets even better. Her paper wasn't just published, it was selected as the editor's choice for that issue. Now, if you're not in the world of science, let me just tell you, that is a huge deal. It's basically the top experts in the field giving you a standing ovation and saying, this research right here, this is not just solid, this is important. And this is where the story explodes, expanding way, way beyond the shrimp hatchery. This brings us to our final section, beyond saving shrimp. The very same ideas that Dr. Shureka used to save those shrimp have massive, and I mean massive implications for human health. This chart says it all. Traditional antibiotics, they're like a carpet bomb. They kill the bad bacteria, sure, but they also wipe out all the good bacteria in the process. Phage therapy, that's a sniper rifle. But here's the most crucial part. As bacteria get stronger and evolve to resist our drugs, creating these superbugs, phages can actually co-evolve right alongside them. We have a weapon that can keep adapting.
And so that's where we'll leave it. A story that started with saving tiny shrimp larvae has unlocked this whole universe of possibilities. It really makes you wonder, doesn't it? As we stare down a future that's seriously threatened by untreatable superbugs, could these microscopic hunters, championed by brilliant researchers like Dr. Surekhamol, actually be our greatest weapon?